Hello, my name is John Kalantari, Assistant Professor at the Mayo Clinic, Department of Surgery, and Center for Individualized Medicine. One of the many uncertainties associated with spaceflight is the initiation and progression of cancer. Molecular alterations are pretty ubiquitous in spaceflight, and they magnify the risk of cancer mortality and morbidity during long-term missions. An example of this is radiation exposure from solar particle events and cosmic rays, which are only a few of the many possible causal drivers of cancer that present a significant health concern for astronauts during interplanetary and future Martian surface missions. Now, by understanding the molecular basis of cancer development, we can better assess the impact of various risk factors on astronaut health and either treat, or better yet, prevent cancer before it arises. The problem is, traditional predictive models only get us so far in allowing us to model what has been seen in our observed data sets. Now, in order to generalize beyond our data and account for the known unknowns of cancer biology and spaceflight, we need to start building new methods of causal reasoning that underlie our predictive models. And to address this gap in knowledge and methodology, the Mayo Clinic is proud to partner with NASA and Google Cloud on the Frontier Development Lab Astronaut Health Challenge. The focus of this year's Astronaut Health project is on the development and application of causal inference methodologies for enhancing our knowledge about the true causal mechanisms underlying cancer evolution. So without further ado, please welcome the 2020 Astronaut Health team.
The human race has always wondered what lies in the universe beyond our planet. In a matter of decades, we will be able to send explorers on the first long-duration space mission. But these pioneers of space exploration will be exposed to extreme conditions. This exposure puts them at a higher risk of cancer. On Earth, cancer touches the lives of millions of people every day. Cancer is not just one disease, but many diseases. And to save lives, we need to build a framework to understand the causal drivers of cancer. A causal inference framework can enable us to pull back the curtain on these complex systems, to understand the mechanisms underlying the most elusive biological and medical challenges that we face, to enable us to break out of our planetary bounds and explore the universe that we inhabit, and to aid us in the search for life beyond our planet.
Welcome everybody. We are the Astronaut Health team, comprised of machine learning and astrobiology wizards from around the globe. We have 4 researchers, Kia Kazili, Adrienne Horfrost, Arno Blas, and myself, Samuel Budd, as well as our 4 fantastic mentors, John Kalantari, Graham McIntosh, Frank Sobachensky, and Kritika De Silva. And over the past 8 weeks, we've been tackling the problem of deciphering the causal mechanisms that underlie cancer. Now, understanding cancer poses 3 unique challenges. Firstly, we have high dimensionality. Cancer primarily occurs due to malfunctioning cells that can exhibit mutations in potentially any combination of the 6.4 billion base pairs in the human genome. And this vast problem space makes the dimensionality of our problem absurdly high. Secondly, the available data sets are very small. Despite the 17 million new cancer cases every year, the biggest consistent cancer data sets typically have fewer than 100 samples. The final challenge is to move beyond finding correlations and to find the true causes or causal drivers that underlie complex biological systems such as cancer. But what does it mean for one thing to be a causal driver of another?
Going back to basics, let's consider the chicken or the egg problem. We can observe millions of chickens and millions of eggs, but this tells us very little about which came first, and whether the egg is a causal driver of the chicken or vice versa. When we start thinking about biological systems like cancer, the questions become a little more complex. Suppose that we find that a mutation to a gene X is frequently observed together with some cancer subtype Y. We might ask, is a mutation to gene X the cause or the effect of that cancer subtype? Or could both be the effect of a third unobserved variable Z, for example, exposure to high radiation? Naturally, there are many such questions that need answering to understand cancer. The importance of unraveling the causal drivers underlying cancer cannot be overstated. By doing so, we can not only develop better treatment strategies, but for the first time, know where to intervene to prevent cancer before it takes a foothold. Acting in this endeavor, we've developed a causal inference framework that is robust to the challenges of biology and cancer. We're proud to introduce CRISP, the Causal Relation Inference and Search Platform. We've developed an ensemble of causal inference methods, from classical approaches to state-of-the-art causal machine learning methods, leveraging the strengths of each to create a virtual petri dish capable of finding causal relationships in extremely complex heterogeneous data. It's designed to be flexible and modular, so it can discover the causal drivers for not just one, but potentially thousands of effects. All that needs to be done is to select the effect to be explained and to provide the data containing both the effect and the potential causes. CRISP then provides the results in user-friendly visualizations to our Streamlit front-end app. We've gone even further and ensured our framework can scale up to the demands of high-dimensional data and multiple queries. Using the incredible resources provided by the FDL and its partners, we're able to ask not just one question, but thousands, and to keep asking questions iteratively until complex biological mechanisms are deciphered. And this is only a prototype. It's ready to be extended toward any stakeholder's desires. So let's have a look at CRISP in action.
As our first use case, we have partnered with the Mayo Clinic to unleash CRISP on the world's largest multi-omic colorectal cancer dataset. Using our interactive front end, we can explore our dataset to build a better understanding of our data. Once our experiment has completed, we can interact with the results to view the predictive performance of each of our methods and analyze their suggested causal features. We can dig deeper into the results and consider how these methods have performed as an ensemble. Here, we can see the combined output of CRISP, view the top overall suggested causal drivers from our ensemble, and use these to deeper understanding of the true nature of the system under the virtual microscope. Of course, it is vital that we validate our findings. However, with the lack of ground truth for our data, this in and of itself is a challenge, which we're overcoming in two ways. We found an overwhelming overlap between suggested causal features from our framework and the efforts of the wider cancer research community. This corroboration of results gives us confidence that CRISP can extract relevant causal features from high dimension, low sample size, complex biological data. But we haven't stopped there. We've conducted extensive experiments on synthetic data that mirrors properties of complex biological data to validate that CRISP has taken us beyond correlation to extract causal drivers and to develop deeper understanding of each of the methods in our ensemble.
So, in summary, what's new about CRISP compared to previous data-driven cancer research? But firstly, we put the focus on causation rather than correlation. This means that in addition to getting an indication of the direction of cause and effect, we're also less likely to identify spurious correlations as causal and more likely to select actual causes as causal. Furthermore, CRISP is a fully modular and easily deployable tool that can help researchers around the world to advance their cancer or biological research. Our code base will be made open source, and it is in fully Dockerized form, making it convenient to run on any data, on any system, at any time, with just a few clicks. So we would like to invite you and take a step forward with us, imagining the year 2035. Imagine the first crewed mission to Mars taking off, with the astronauts knowing they're safe from cancer, as its occurrence and development have been fully understood and can be prevented. Imagine the up to 17 million lives per year that will be saved here on Earth. We believe that this vision can become true and that we should all endeavor to make that happen. We hope that automated causal discovery guided by CRISP can help us all to achieve it. So we would like to thank you all for joining in today. And at this point also would like to thank again all our partners around FDL for making this research possible. Thanks.
Thank you, Astronaut Health Team. And I'd like to go ahead and invite our panelists back. So this time I'd like to know from each of you, what are the implications of this remarkable work that this team has done? And so to get us started, I'd like to turn this over to Aaron.
Yeah, that's remarkable implications. We could, I mean, for the obvious, definitely extend this knowledge to cancer populations across the earth, but particularly the implications would be greatest felt in low-income countries.
Great, thank you, Aaron. And now I'd like to turn it over to Rodney.
Thank you, Sarah. So yeah, there are obvious implications here. This is moving, I think, the needle of the recruitment of AI ML techniques to support dual use, what I like to call dual use capabilities or technologies. So clearly, crew members who are going on long duration space flight excursions are going to be exposed to environments which make them a bit more vulnerable. But the fact that we can actually close the loop back to inform how potentially, those insights can support terrestrial diseases of a similar or the same nature is extremely important and valuable. So that's one of the clear implications, and one of the reasons why this particular challenge, among others, I think is particularly well suited to be able to address those for cancer and a lot of other terrestrial diseases.
Wonderful, thank you, Rodney. I couldn't agree more. And Scott, to close us out.
Hey, Sarah. What I love about this work is that it's a hard problem. How do you know this? The sources of a cancer. We have this not only for the space exploration, but here on Earth. And what they've done here is they've married AI with fundamental how we do cancer research, the science of that, and they made it, and they're making it as easy as ordering like a crispy chicken sandwich at Chick-fil-A, right? The idea is that if you have that kind of tool, if it's that easy and that accessible, imagine what all the scientists can do in all disciplines. And I certainly would I like to see Crispy right on the launch pad with them as they're going the mission and help understand what are causal effects, because the mathematics is so complicated for the uninitiated. But to make it that accessible, I think this is the sort of trend we're seeing in tools. It can really, as a race, start to the human race. How do you put together these great tools with the human mind and do things we could never do before?
Thank you, Scott. Yes, I think accessibility is key from the work that this team has done. So with that, I'd like to say thank you to the Astronaut Health team and thank you to our panelists. We're going to go ahead and move over to our two heliophysics teams. And to introduce them, we have our star, Lika, and let's go ahead